Assalamu alaikum warahmatullah. I hope all of you will be well, and you are watching my YouTube channel Nature and Science with Abdullah, and my name is Muhammad Abdullah. I am here with a new topic with you, with a presentation on a topic understanding chromosomal substitutional lines and chromosomal segment substitution lines in plant genetics and breeding. So I belong to a department of plant breeding and genetics from University of Agriculture, Faisalabad. So this presentation is being delivered on 25th April 2024 on World DNA Day. How DNA revolutionized the word not only in agriculture, medicine, but all aspects of molecular biology that are being utilized in life sciences. So moving forward, in this presentation we will cover introduction about our topic, and which is chromosomal substitutional lines and chromosomal segment substitution line, and then developing methodology of these line and application of these CSSLs and CSLs lines in this presentation. If I talk about introduction, plants inherit their traits through chromosomes. They pass their phenotypic traits through their genetic material, which is present on chromosomes, and chromosomes are present on nucleus. Chromosomes are thread-like structure that carry genetic information from one generation to next. Genetic research and crop improvement programs in the world rely heavily on development of specialized plant lines or plant genetic material that allow for the systematic dissection of complex traits and identification of important traits. Because if we have a diversity in our germplasm, or other than that, if we have some specific material which will be feasible for us to dissect the genetic makeup of any trait, so nowadays breeding and genetic goals are depending upon these new materials or germplasm resources. So two such type of valuable resources are chromosomal substitutional lines and chromosomal segment substitutional lines. What are these lines? Let's come to discuss. These lines are developed, which are chromosomal segment and chromosomal segment substitution lines. These lines are developed by crossing a well-adapted parent, which is present already in the market as a recurrent parent, with another parent as a donor parent. The second parent that will be utilized as a donor parent, this parent will be used for transfer of a particular trait that was lagging in our recurrent parent, or we can say that was lagging in our well-adapted parent. So most of the time, the donor parent that are utilized in this in the development of these lines is mostly belongs to a wild relative of that crop. So another parent will be another parent which will be donor parent will be wild relative of that parent. So in most cases, as I discussed, we use second parents from its wild relative of that crops. So for preparation of CSL lines, which are chromosomal substitution lines, it involves the substitution of entire genome of a donor parent into the genetic background of a recurrent parent. When we develop these lines, the chromosomal parts or chromosomes of donor parents are completely substituted from the chromosomes of recurrent parent in the lines which will be being developed. This creates a set of lines, each carrying a different chromosome or chromosomal arm from the donor parent into the recurrent parent population at the end. In contrast, CSSL lines, which are chromosomal segment substitution lines, it involved the substitution of specific segments from the chromosome of a donor parent, which I discussed, which will be wild relative, into the genetic background of a recurrent parent, which will be our adaptive variety. CSSLs will be the set of lines where each line contain a specific chromosomal segment from the donor parent introgressed into the genetic background of a recurrent parent. It will be easy when we will see its systematic diagram to understand. So moving forward, now I will discuss the developing methodology of these lines. First, we talk about chromosomal substitution lines, in which whole chromosome will be substituted from the donor parent into the recurrent parent, or we can say a chromosomal arm, not less than from chromosome arm. Our chromosomal arm will be substituted from the recurrent parent by introduction from donor parent. So for development of these lines, first we need two parents. One, as I discussed, recurrent parent, and second one will be as our donor parent. Then initial hybridization, we perform crossing between both parents. Then there will be back crossing, multiple time back crossing for the integration of our because we want to integrate the most genetic makeup from our well-adaptive variety. So we will perform multiple backcrossing with our recurrent parent. Then marker-assisted selection will be a key tool, and cytogenetics analysis, like in the form of fish and gish, that will be utilized time to time to analyze our results to select substitution lines initially. So at the end, we will evaluate our lines and characterize the lines, like which line contain which chromosome from donor parent, because when we attain lines at the end, each line will have different chromosome. Like one line will have chromosome 1, and second line will have chromosome 2 substituted from donor parent, and third line will contain chromosome 3 from donor parent, which will be substituted in the recurrent parent. So here is the systematic diagram. Look here, I took the example of wheat here. So triticum estimum is a common weight which is 2x is equal to 6x. Hexaploid in chromosome is equal to 42. This is well adapted variety. This is well adapted hexaploid genome of wheat, which is being utilized all over the world. So this is very suitable for growing, for harvesting, for utilizing. But sometimes what we see, we lag behind one or two traits of, like in the form of disease resistance when new disease came, or any type of another traits that could be lacking in this line. So in this case, what we have to do, so in this case we cross our triticum estimum with wide relative of hexaploid wheat, which is Agelostoschi, a diploid parent in which is also present in subgenome of triticum estimum as a D genome. So we will cross our hexaploid triticum estimum with Agelostoschi to transfer some traits which are required in triticum estimum. So we will first of all we will cross them, and we will attain F1 generation. Then this F1 generation will be crossed again with triticum estimum, because we want a very little genetic material from Agelostoschi and more genetic material from triticum estimum, which is a well adaptive variety in the market. So after crossing F1 with triticum estimum, we will attain backcross 1 F1 population. So after attaining backcross 1 F1 population, we will again cross backcross 1 population with triticum estimum. And before making cross, we will select those lines based upon marker assisted selection. We will perform marker assisted selection on this germplasm, which will indicate us these lines have some genetic some chromosomes from Agelostoschi, which was donor parent, or not. So this will indicate that we have some chromosome substituted in our backcross 1 population. After making selection based upon marker assisted selection, we will cross again it with triticum estimum to attain backcross 2 F1 population, because it was backcross 1 population, and it will be, after crossing with triticum estimum again, it will be backcross 2 F1 population. Again we will perform marker assisted selection to select those plants which have substituted chromosomes, and this backcrossing we will perform multiple times. Continue backcrossing will be performed with triticum estimum, which is a recurrent parent, and at the end of multiple backcrossing, we will attain backcross population 5 or more than 5 or 6, but this will be F1 population because we did not perform selfing yet. So after attaining a higher level of backcross population, we will perform selfing, because we want to fix the genetic material that we have attained from Agelostoschi. So creating we can say that for creating homozygosity in our germplasm, we will perform selfing, and we will develop backcross 6 population with F5, because multiple selfing will be performed. So our population that will become at the end will be backcross 6 F5th population, because selfing will be performed 5 times. This depend upon your breeding goal, your resources, your time, and your what you want to study, your goal. So this whole scheme can be changed. Then at the end, when we will attain our backcross 6 F5 population, we will again perform marker assisted selection to select those lines which has substituted chromosomes, and we can also perform some cytometric techniques to analyze the chromosomal pattern in the transfer in the substituted lines. So some techniques which are very common in cytogenetics, like in current era, fish and gish, these techniques will help us to identify which chromosomes are transferred are substituted into the recurrent parent from donor parent. So these techniques are can be performed in chromosomal substitutional lines, in developing chromosomal substitution line. At the end of this uh, whole scheme, we will attain chromosomal substitution lines in the field that can be utilized for future purposes. So this is the methodology for developing chromosomal substitution lines. We have discussed one part. Moving forward, if we want to develop chromosomal segment substitutional lines, CSLs, the procedure is same with little bit changing here. We will select parental lines. We will perform initial hybridization between them. Then multiple back crossing will be performed, as I discussed before, in CSL lines. Then we will perform marker assisted selection and evaluation and characterization of these lines in the field. But there is a little change. Here we are not performing cytometrical analysis, cytogenetics techniques. Why? This is the question. Why we are not performing these techniques which we have performed here, cytogenetics techniques? Because in this methodology, we were developing chromosomal substitution lines. Entire chromosomes were being substituted. But here we need to transfer only segments, not only complete chromosomes. So these cytometric techniques that are available in the market, like fish gish and many other improved techniques, are not able to to dissect the minute chromosomal segments that will be transferred from donor parent into our recurrent parents. So we will not perform cytometric techniques, which are because it is not possible for cytometrical analysis oh, to to catch those regions which are being segments, because they are much smaller in size than large chromosome size. So for developing these lines, we will follow the same methodology. We will take triticum aestivum, we will cross it with Agelostoshae, then we will develop F1 will be crossed with triticum aestivum, then back cross 1 population will become. Then again, after multiple back crossing and marker restricted selection, we will collect back cross 6 population with F5, because selfing will be performed to fix the genetic material for developing homozygosity in our lines. Then at the end, we will select those lines which has chromosomal segment substitution lines. Marker restricted selection will tell us which parts of the uh, chromosomes are being transferred in our germplasm. So this is the second methodology to developing uh, chromosomal segment substitution line. This methodology is not much different from chromosomal segment substitution lines, which I have discussed here, but little change. We did not perform cytometric analysis here, because chromosomal segments are very small in size, that chromosomal cytometrical and techniques are not able to detect these small size of segments. Moving forward, if I talk about the application of chromosomal segment substitution lines or chromosomal substitutional lines, we can utilize these lines, which will be developed at the end of whole process, which will be chromosome substitution line or chromosomal segment substitution line. These lines, we can utilize these lines for fine mapping of QTL's quantitative trait loci, mean to say which part of the genomes in an which part of the genome in a plant are controlling a particular quantitative trait. We will be able to easily dissect. Then after QTL development, we will be able to discover genetic material or gene that are controlling a particular trait very easily. Then trait mapping and further genetic analysis will be possible. And moreover, we can introduce the desired traits, because we already performed here characterization of our germplasm, like which lines here is example. These are the lines. Think about that. These are the lines which are chromosomal substitutional or chromosomal segment substitution lines, and we have characterized that this line have rust resistance, and this line have drought tolerance. This line have heat tolerance. These lines, we will be able to utilize these lines in future for transfer desirable traits through integration again. So these are the application of CSLs and CSSLs and CSLs, which are chromosomal segment substitution lines and chromosomal substitutional lines. So this was all about, and these are the reference you can further study from these resources, and many other resources are also available on PubMed, Google Scholar, and many other such resources databases. So thank you very much for listening my presentation. I hope this little effort will be valuable for scientific community to transfer or developing such lines. Thank you so much for listening, and Allah Hafiz.